Hi, everybody. So I just want to thank everyone for joining this presentation this Wednesday morning. I want to be respectful of everyone's time, so I also want to get to the fantastic content we have. So I'll keep the introduction super quick. My name is Justin Liu. I'm the Global Marketing Lead here at Azure, and I'll be moderating today's session. So please feel free to chat any questions in the box on the bottom right-hand side. You can also find the chat function on the bottom tab. It's like the little speech bubble. So we'll have a Q&A session at the end. So please chat your questions, and I'll be passing them along to Brian either throughout the presentation or at the end. So please feel free to test this function now by saying hi in the chat. So I'll just say hi to everybody. Feel free to respond and say hello back. Additionally, at the end of the presentation, we'll be revealing a special coupon code to our website. So make sure to stay until the very end of the presentation so you guys can use that. So without further ado, I'll introduce today's speaker, Dr. Brian Bauereis. So he did his PhD study at UTHSC in San Antonio, Texas, and then his postdoc at Yale University in New Haven, Connecticut. His postdoctoral research focus was on Parkinson's disease, and specifically the function of dopaminergic neurons in the basal ganglia. So in this presentation, he's going to go over Azure's advancements in imaging technology and pretty much why he wishes he had one in his lab. That was a joke. You guys can laugh. And now
very clever, Justin.
We'll go over.
Thank you, Justin. I really appreciate it. Welcome everybody to today's webinar. As Justin mentioned, we'll be talking about the Sapphire biomolecular scanner. Today's agenda will be focused on who we are as Azure Biosystems. Next, I'll provide you with the inside scoop for the technology inside the Sapphire. And then lastly, can your Western blot imager do this? We're going to go way beyond just the standard blots and fluorescence and chemiluminescence to really showcase what the Sapphire and even our other instruments are able to do. So at this time, I'm going to go ahead and turn off my video just so it's less distracting. And again, as Justin mentioned, please, if you have any questions, throw them in the chat and we'll answer them at the end of the presentation. So Azure Biosystems, we simply do not employ sales professionals. We hire scientists that understand how science and technology go hand in hand. This actually enables our team to be able to assist with scientific protocols, provide instrument maintenance when necessary, and even fully understand the complex scientific experimentation to better assist you guys, our customers. So we work hard in order for you to have the best data possible to keep your research flowing. We are the only company that does not manipulate data. We are true scientists that respect the scientific method and understand what journals require and the trends associated. There we go. So Azure, we do have a little bit of a history of imaging. We are based in California. All of our products, instruments are manufactured in the USA, right there in California, in the San Francisco Bay Area. So our team of engineers have been together for over 2 decades now. Originally, they started with Alpha Initech back in 1992. You can see some of the stats of the number of systems that went out just in that time span. The company then did morph into Protein Simple, later to Applegen in 2011. Protein Simple decided to go along the route of more capillary electrophoresis, liquid chromatography. And so even though they still supported some of the instruments that actually came out during that time, their focus was less on Western blotting instrumentation. So our chief engineers decided, well, this is a great time for us to go ahead and start our new company, Azure Biosystems, back in 2013, to address all of the issues that have been present with the instrumentation back in the day. And I think we did an excellent job with coming up with some technology, which I'll be able to point to throughout this presentation. So today is going to be a discussion of the Sapphire. This is our laser biomolecular imager. You could actually get it in 3 different models. So either you could have just the Sapphire NearIR, which would only have the 2 lasers for near-infrared fluorescence. The Sapphire RGB, that gives you the red, green, and blue fluorescence, so 3 lasers. Or you can get the full package with the RGB NearIR, and you have the 4 full solid-state lasers. Additionally, for each model, you can include chemi imaging and phosphor imaging. So this is truly a revolutionary instrument. Nothing like this exists currently on market, and it is literally the best of the best. And I'll be able to explain why that is as we go on. So the evolution of excitation sources, it started with obviously white light. From there, you can apply excitation and emission filters, where you can actually then focus that a little bit better to the color that you like. But as you can see from the very bottom, the signal to noise ratio was quite low. Signal was there, however, the background noise was quite high, and so it made it difficult. From there, the progression of technology went to LEDs. LEDs are much better at providing more optimal light in the correct color range, again, with the excitation emission filters. You definitely still have some signal to noise ratio, but you can actually get much better data. You can even quantify that quite easily. The last is the infrared lasers. So, this came out quite a long time ago, and the technology for the lasers has only increased since then. Obviously, it's superior. I don't think I need to explain that to you guys. I mean, it's a direct beam, collimated light. The signal to noise ratio is extremely well. I mean, look at that background there on the high. It's almost non-existent. And that's what you get with lasers. So now you can see how our performance stacks up to some of the more similar instruments, and even one of our own. So if we look here, I'm sure you guys recognize the LightCore Odyssey. They use an APD detector. That's an avalanche photodiode, and they focus specifically in the near infrared range for fluorescence. If we look over at the GE Amersham Typhoon, they use a PMT detector, which is a photomultiplier tube, and they can do visible NearIR and phosphor imaging. Now, if we look over at the Sapphire, and then beneath that is our other product line, it's a CCD imager. So it uses a CCD camera that can do ECL, chemiluminescence, it can do RGB with the LEDs, and then near infrared is actually with the lasers. Now, because we use the LEDs and a CCD camera for the system, it's not the true power of a full laser system, which is why we created the Sapphire. There was really no competition to exist for the Typhoon. And so now we've created this Sapphire that has all three detectors in it. It has the APD, it has the CCD, and the PMT. And I'll get into that. We'll dive a little bit more deeper into that in just a moment. So some of the compatible fluorochromes that you can get for this is not an exhaustive list. If it was an exhaustive list, we would probably spend quite a good deal of time discussing on everything. Our systems are, I call them reagent agnostic. Most reagents from any vendors will work on the Sapphire or even our other product lines. So now we're going to get back into the kind of unique detection design of the Sapphire and why we chose the specific detectors like we did. So just to reiterate, the Sapphire can do phosphor imaging, fluorescent imaging, and chemiluminescence. So what we had found that with the photomultiplier tubes, which you can see for number one, this gives excellent quantum efficiency in the UV and short visible range and low QE in the NearIR, which is why high QE for the photomultiplier tube, which means it's going to be great for phosphor imaging, as well as the initial visible light spectrum to include the blue. That photodetector does a great job for that. For fluorescent imaging, the avalanche photodiode has very high QE in the NearIR, which is why we use it for specifically our near infrared channels. And of course, chemiluminescence, the only way to have true chemiluminescence is to have a CCD camera put into the system, and that's exactly what we did. So we did identify the best detectors to go into the system, and now we have all three of them in there. So you can see the dynamic range is 4 to 6 orders of magnitude for 1 and 2, and then for the CCD device, it's between 3 and 4. So why 3 detector types? So as you can see here, this is an excellent graph looking at the quantum efficiency over the wavelength. And if you look at the red dotted line, that's the actual PMT, and we're looking at such a far out wavelength that this is the NearIR. You can see how that drops significantly once you get into that NearIR range at about 700 nanometers to a little bit beyond 800. However, if you look up high at the green line, that's the APD. Way better quantum efficiency. So anytime that you guys are doing in the NearIR range, you definitely want to make sure that you have the APD on board for that, which is, of course, the Sapphire. So you can see some of the scanner details. beneath that. So obviously it's multiple independent detection channels, so a multiplex reaction. 4-channel sampling in each scanning step. So only one scan. It'll do simultaneous scanning. So gone are the days of waiting forever to get an image from a scanner. You have a 25 by 25 field of view. Laser and detector settings are combined into the capture software, so you can adjust for sensitivity, focal plane settings, and even the 10 micrometer max resolution, which is just a beautiful spec to have. And I'll be able to give you guys some graphical representations of what 10 micrometers can actually see. Next to that, of course, the CCD. As I mentioned, it's great for chemiluminescence. So that's the reason that we put it in there. You can simultaneously detect light in a big area, so it's a single scan. The field of view is 16 by 13. You can change the sensitivity by altering the pixel binning if necessary. And of course, you can expose up to a full hour, which is highly unlikely. Our systems are the most sensitive and largest dynamic range systems in the market, so it's very rare for something to take that long. So going back, dynamic range, it does depend on the detector. So dynamic range is simply the ability to capture both very low and high signal intensity in the same image without saturation. To break that down into simpler terms, it simply means that you can get either, if you have a very low abundant protein or a very high abundant protein, the chances are that you would get it within a linear range on our systems is high. So it's very easy to quantitate on our systems versus others. So you can see the dynamic range is quite different for these 3 different types. Film, it's kind of the older technology. If you guys have questions about how this stacks up to film, we can discuss it at the end of the webinar. And then, of course, you've seen the 3 to 4 ODs for the other 3 that are put into the Sapphire. So with the actual system, you would be getting a computer, and the software is to be included. So the workflow is so easy for gels, blots, literally everything. And I have a few slides that I can showcase what it actually looks like. But it's the application-driven software that makes it very easy to set up. It walks you through exactly how to image. This should not be a difficult thing for researchers. The software should be easy to use and very intuitive, and that's exactly how they created it. So this is actually looking at the capture software itself. You can see that the first red rectangle in the upper, almost in the middle, but you can see on imaging it says fluorescence, chemiluminescence, and visible. That's simply where you would choose what type of experiment you're about to image. If we scoot over to the right, you can see that you can change the resolution going down to 10 micrometers and all the way up to 1,000 micrometers. But even more importantly, and really cool, is you can control the focal plane by the software itself. So if you look at the small rectangle box on the right, you can see that you can drop it down. I forget the exact increments, but I believe you can change the millimeter spec. So it's very similar to a confocal, but without Z-stack. I have some images that I'll show later on that has this built-in capability, so you can actually see what we're talking about. Next, imaging for chemiluminescence, very simple. You can see that there's an image capture button on the right. You can do single, cumulative, multiple. It's all just laid out for you. You can even see there's a marker button that's on the bottom right. So it'll actually take an image of your marker, and you can combine those 2 together automatically. So it removes a lot of the guesswork with ladders. And as you can see here, this is the gallery. So this is completed 4 images here, and it just looks super crisp and clean. Of course, you can change the contrast, all of the other settings that you can see there on your screen. So let's talk a little bit about performance. How does this stack up to the other known instruments out in the field? And it's on par or better. So this one, we're looking at RGB fluorescent performance. So we're looking at the sensitivity, linearity, and dynamic range of the Sapphire versus the Typhoon. And as you can see, they are just neck and neck with each other. We have slightly better dynamic range and maybe just a couple decimal points different in signal linearity, but they're very similar. NearIR fluorescent performance, again, very similar. We do have a little bit of an advantage on the signal linearity for that. ECL performance. I mean, you can't image chemiluminescence on another system. It's the Sapphire that you can do this with the full laser system. And the sensitivity equals or exceeds film detection. So if you just take a quick glance down here, you can see that, I mean, look at that, that line. So we have the plots graphed out for the Sapphire biomolecular scanner versus the film. And then you can see on the right, you can actually put those samples together with the weighted markers, do that automatically, and it looks beautiful. phosphor imaging performance. I won't spend a lot of time on this, but you actually, if you want to have that as your module, you can even do phosphor imaging. And this is a beautiful 35S page gel that was taken. You can see how crisp and clean it looks. Gel imaging performance, look at that DNA gel. It's gorgeous. And then we have a Kamasi gel just to the right of that. We also can do any of the other protein gels as well. These are just examples. We can see that our resolution, we do go down to 10 micrometers and of course all the way up to 1,000 micrometers. This is just a simple full moon biocalibration slide, but you can see that obviously resolution does play a big role, especially if you're getting outside of the blot that we're going to be talking about soon. Okay, so now we're going to go into a little bit of the applications. This is where it's going to start getting a little interesting. So we'll talk about all of the different components that you see on your screen there. So fluorescent applications, obviously it's a fluorescent imager. These are just some examples of what can be done: membrane, cell-based assays, tissue section, molecular imaging, gels, in vitro. The list goes on and on. As you can imagine, if you're able to get a fluorophore with a specific antibody, you can use the Sapphire in almost an unlimited potential. So I wanted to throw this slide in because I wanted to address that a lot of researchers believe that fluorescence is very finicky, difficult to work with. There's extra steps, but actually none of that is really true. It follows exactly the same protocol as for chemiluminescence, but if you notice, just versus the fluorescence, the only thing that's different is you don't have to do the ECL detection reagent. You just literally conjugate your secondary, and then boom, you look for the fluorescence. So you can image and analyze on the Sapphire or any other system for that matter. It's exactly the same, only this data is easily quantifiable and it doesn't have to be justified. It's essentially one photon in, one photon out. That's the beauty of fluorescence. So this is a great slide just showcasing what a multiplex reaction would look like on the Sapphire. So we have fluorescent detection of STAT1, which is your Cy3 or the blue dye. We have the phosphostat1 as the IR700, the red, and then GAPDH is the IR800. Combine them together and it looks beautiful. So you can actually detect the phosphorylation of that protein just with that slight molecular shift. If we were to do this in chemiluminescence, you might be able to see those blue bands next to the red, but it would be quite difficult. So this is, again, where fluorescence is king. So power of 3 lasers, what does that mean? Quantitation. So if you choose the system that has the RGB, the 3 layers, what you can do is you can detect either 2 proteins and normalize to a housekeeping protein loading control. You can detect 2 proteins and normalize to a total protein stain. And you can even detect 2 proteins and normalize to a total cell stain. So these are gorgeous images. And now what you guys have to keep in mind is this is kind of the shift for the standards. A number of researchers had noticed that the housekeeping proteins can be altered under various experimental conditions, and therefore it is not the standard to be used in those situations. And so Azure and other companies have come out with total protein stains that you can use in conjunction with your probing of your specific antibodies. So that way you don't have to strip, reprobe. It's all done on one thing with 3 lasers all at once. It's a beautiful thing. Now, this is the types of quantitative analysis, just like I was just showing you here. So you could do the HKP normalization of the ratio of A to B. You could also do the ratio of C to B. And you can even look at the total protein stain normalization. So the calculation of ratios to account for that variability in both sample loading and transfer efficiency. And this is a beautiful image that we've captured on the Sapphire. So data comparison should only be made from comparative experiments, and individual data should not be utilized across multiple figures. I think that's pretty standard. I think most of you guys realize that journals do require that standard be held. Now I'm going to talk a little bit about immunofluorescent plates. So what you could do, let's say that this is you're probing for 3 different antibodies. You could use the 550 laser, 700 laser, 800 laser, and then you can triplex and overlay them. So a way to quantify this, so one thing that you could do is you could do an in-cell Western. So essentially you have your cell cultures you can treat with inhibitor, stimulator, etc. You can fix the cells, permeabilize, incubate with your primary and secondary, and then scan directly onto the Sapphire. Alternatively, you could do on-cell. not in cell, very similar, except you skip the permealization step. You could even do a suspension of the cell culture slides, or you can even do a live version of it. These are, again, just options that you can consider when doing Western blotting techniques. Sometimes this you could do many different samples in triplicates and have way more data than a single blot or gel could give you. Something to keep in mind. Again, you can even normalize against a specific DNA stain that had been done in this slide here. Now we're going to go on to a little bit more exciting and visually intriguing slides. So the beauty of the Sapphire, 10 micrometers resolution, you can really get down and identify very specific things. So we're looking here at rat brain slices. Now we're looking at tissue sections. So these are actual motor neurons. So the cell body of a motor neuron is approximately like 100 micrometers, whereas the diameter of the axon is about 1 micrometer. If you look real closely at your monitor, you can actually make out some of the axons that are coming out of these individual motor neurons. So again, this is a great example of what can be done on an instrument, that was originally built for Western blotting, but because it has such great optics, excellent performance, you're able to get results like this. And so I do recommend to the Sapphire users, if they do IHC, where they have many slides of either tissue sections or something of the similar, you can actually put them all onto the Sapphire, image that, and that could stay in your notebook as your designation versus some arbitrarily written thing on a glass slide that you may lose later. So it's not a microscope. However, it will give you that broad general picture so you can put them all together, and that could be your documentation for your notebook. Proceeding, we have whole organ sections. So this is a stem of a paragonium using a 10 micrometer resolution again. Looks beautiful. This is a GFP plant. So even botany is getting some recognition. Here's whole organ sections. I love this image. So you can actually see small features down into the tissue section. So this sheep kidney was stained using the quick stain reagent. Whole tissue was scanned at 488 nanometers and 658 at a 20 micrometer, 50 centimeter per second scan speed. So this takes it down to 20 micrometers. So we could even do better than that, and you would be able to make out individual cells. Next, we have a cow stomach. So you can see the prep to the left there, cow stomach on the right, and then I have a blown up image so you can see greater resolution. There's 10 micrometers for you right there. If you were to do this on a microscope or even a confocal microscope, it may take multiple images that may have to be stitched together depending on the size of the tissue. So if you guys are doing any sort of tissue imaging, whole animal models, the Sapphire is a perfect tool to use in conjunction. So you can even do molecular imaging, so you can identify in vivo phenotype specific targeting. This is going a little bit beyond the range of sticking with tissue sections. However, all this stuff is possible with the Sapphire. So why NearIR? There's a lot of reasons why many researchers are moving beyond the typical RGB section and moving towards NearIR. There's actually many reasons for this. So it's less light absorption. So you can get deeper excitation light penetration, easier emission light detection, and of course, using laser excitation, you have the highest energy. You also get less light scattering. So lipids are better visualized. Low autofluorescence. NearIR dyes are not hindered by tissue autofluorescence. And so they're ideal for any sort of application that you want to look at anatomical structures. It's important to remember. NIR is definitely the new trend. So some other molecular imaging research areas, as you can see, this is again not an exhaustive list. As I go through the slides, you'll get more and more list. But as you can imagine, if you're working in any one of these general categories, you can identify specific experimentation that the Sapphire could be utilized with quite beautifully. So even just looking at tissue growth monitoring, effects of drugs, tumor physiology, tumor vasculature, all of this stuff is possible with the Sapphire. You can look at diseases, translational research, of course, inflammation, organ and tissue transplantation, cell transplantation, cell replacement. This is what I spent 4 years at Yale doing. I would have loved to have this instrument, as Justin was teasing me about. It would have helped tremendously because I had to use my tissue sections on a microscope that had to stitch all these images back together when I could have had the entire image taken once with that 10 micrometer resolution. It would have helped tremendously for my research. So hopefully I can pass it on to you guys and you can use it for your research. Gene therapy, viral vectors, non-viral vectors, all this stuff can be used with the Sapphire there. So what can be detected? So it's important to point out, so you can see the lid open up and you can see the glass field of view there. There is room between that lid and scanning the glass to image a mouse. You can put other objects in there as well, three-dimensional objects. And remember, you can change the focal plane, which means you can create a pseudo Z-stack. And I'm getting very close to those images. Just hold tight and we're almost there. So targeted approaches. So you could use gene-encoded proteins, GFP, RFP, NearIR membrane labeling reagents. That's a possibility. Injectable NearIR labeling agents. Again, another possibility for targeted approaches. Some more of those gene-encoded fluorescent proteins. Obviously, this is referring to GFP, RFP, Neptune. We also have phytochrome-based fluorescent proteins. So you have biliverdin and cherry. All of these work beautifully for labeling very specific tissues. My apologies, there it comes. So this means that you can use it as protein tags, reporters of gene expression, cell lineage tracking, even stem cells, and proteins for deep tissue imaging in mammals. So track the movement of various molecules or even do in vivo proliferation assays. Okay, so a gene-encoded protein. So this is going a little bit more in depth into some work from Roger Sin. So in May of 2009, he is the Nobel Prize winner for his GFP work. He actually published some exciting work on a new infrared fluorescent protein. This protein is engineered from it's a bacterial phytochrome incorporating the biliverdin as the chromophore. So it emits about 700 nanometer wavelength. And so his paper also demonstrated the challenges with GFP and the benefits of moving to an NearIR fluorescent protein. Again, for the exact same reasons that we just discussed, why NearIR might be better versus like GFP or any of the visible range proteins that we're used to working with. So if you can look here on the left figure, you have the 85I transfected mouse that expresses that IFP and GFP. And then beneath that, you have the 85K as a transfected mouse line that expresses MCATE, which is a fluorescent protein that emits around 635 nanometers. So his group found that a strong signal could be detected with IFP. GFP could only detect tissue autofluorescence. So if you look just on the left graph over here, if you look at the GFP channel, which is on the right of that box, it autofluoresces like crazy. However, if you take it back to the IFP channel, you can see that you do see that bright resemblance in that liver there. So I'll go down, change this over, liver exposed. Look at how clear that IRFP shows up now. So this was back in 2009. I'm honestly a little bit shocked that more people are not using this near infrared reporter versus GFP, but I think that so many scientists have gotten used to using GFP, including myself, that it's just become commonplace. But I do invite you to check out some of the near infrared dyes. They might do actually a better job at visualizing your research versus maybe what you're currently using. And just for reference, if you guys want to check it out, there's the paper where they describe that new infrared fluorescent protein for in vivo imaging. So please check that out at your convenience. So just another option that you can do the aliphatic tails and fluorescent dyes. So this would be great for short-term in vitro proliferation studies. And again, cell tracking, whether it's in vivo or in isolated organs. It's another option for throwing in that secondary where you might want to use it and get some beautiful data. You can also do densitometry. So you can even accurately quantitate the subtle changes in protein expression. So now we're looking at this image. This is a 2G application. So helolysate was labeled as Cy3. Treated helolysate was labeled as Cy5. They were simultaneously separated in the IEF in the first dimension and then SDS page in the second dimension. And that looks really good. That is an excellent result. We can also do in-gel westerns. As you can see, there's no transfer blocking. You can do 2-target detection. It's ideal for detecting hard to transfer proteins, and it's a qualitative detection. You can even look at transcriptomics. So if you look at the electrophoretic mobility shift assay, if you guys are unfamiliar with this, it's a very simple experimental protocol. Feel free to look it up in your free time. However, the NearIR labeled DNA and protein extracts are mixed. Then that shift of the labeled DNA occurs once the protein binds. And then as your controls, you can see for the unbound oligos, for specific and unspecific ones. And you can actually see the shift occur in the protein DNA complex. So this could be a useful experiment to showcase what's happening from the transcriptome level and then the actual data that was found in the paper. Protein arrays. I mean, this is, again, another full moon calibration slide assay, but this is at 10 micrometers. So just imagine if that was your raw data in there, it would look that sharp and clean. You can do microarrays even at 10 micrometers. That looks absolutely clear, crisp, and beautiful. And then we have the FIFA. So this is the NearIR fluorescent immunofocus assay. This was from a paper that I found where they're trying to identify a better way of quantifying non-cytopathic or minimally cytopathic viral infections. And so as you can see, the older technique of using the radioactive IFA is on the left. The newer one is the IR FIFA technique. And as you can see, I mean, just from the resolution standpoint alone, you're getting much more detailed information. It is quantitative as long as you fix before removing the semi-solid media. It's definitely superior over the RFIA methods, which are seen on the left of that image. 6 days versus 7 to 13, and the option for simultaneous detection of 2 targets is there. And it's, of course, optimal for tiny plaques. So I wanted to kind of conclude with this image here. So last year, one of our technical product specialists, Dr. Andreas, he had taken a lot of images on the Sapphire to include this honey melon image that he submitted to the WikiScience competition in 2019, and we actually won that. So it's as you can see, it's just a gorgeous image, and it's super crisp, and not surprisingly, we did win that. So thank you guys for putting up with me and listening to me for 30 whole minutes. You know, I love this humor slide at the end. You know, ever make mistakes in the lab? You know, I certainly did. Let's make them birds. Yeah, they're birds now. So if you ever make mistakes, keep in mind it's fun to make fun of yourself. Western blotting is not always an easy thing. It's a very classical experimentation method that is typically handed down to undergrads, grad students, postdocs. But once you perfect it, boy, it is an artwork. It's an art form. And I feel like everyone could do a great job with it. So if you happen to come to your Azure rep, if you guys want to demo our instrumentation, we'd be happy to do that. We provide demos in the labs all the time. We can even do it now amidst this COVID pandemic. We do come with all our PPE on. We wipe down the instruments both before and after, and we maintain social distancing. So if any of you guys want a demonstration of either the Sapphire or any one of our products, reach out to myself or Azure Biosystems, and we can set that up for you guys. So as Justin mentioned in the beginning, we do have a coupon code. If you guys want to write this down or go into Azure Biosystems and put some stuff in your cart for our reagents, we'll take out 30% off. This will last until April 1st, so please take advantage of this while you can. We also have a webinar feedback form. If you guys can scan this with your camera, you're able to provide us back some feedback, whether that be options for future webinars or anything of the such. And so now we'll take questions. So I will come back on with my video. Hopefully Justin is still here, paying attention so I can find all these questions. Justin, are you available?
Yes. Hi, everybody. So we had one question early in the beginning asking if this presentation is going to be recorded. The answer is yes. It was recorded. We're going to stop the recording now that we've come into the Q&A.